This is the lecture for Chapter 4, Cell Structure and Function. As I mentioned in the first chapter, science gets information about the natural world and then tries to organize that information in a logical manner. And this image shows you how that can be organized from the very small level to the very large level. And in the last two chapters, we focused on chemistry, which is the study of atoms and molecules. Now in this chapter, we are going to focus on the cell. So this chapter is going to look at the structure and function of a basic cell. The cell theory is a very important theory to biology. And remember that the term theory means something, an idea, that is supported by a wide body of experimental evidence and observation. So the cell theory is something that is accepted as fact. And the cell theory is made up of three basic tenets. The first one is that all organisms are made of one or more cells. So on Earth, all the living things that we come in contact with, they must be composed of cells. The second part to the cell theory is that the cell is the basic unit of all living things. So basically, the cell is the smallest structure that has the characteristics of life. And the third part of the cell theory is that all cells come from existing cells. So basically, cells are self-reproducing. So there's no such thing as spontaneous generation, the idea where if you bring non-living components together, you can just spontaneously create a cell. Cells can only come from pre-existing cells. This is a diagram that relates the different sizes of living things to each other. And on the left, it has very small measurements, and on the right, or as you go to the right, it gets to very large measurements. So the largest organism that is on this diagram is the blue whale, and the smallest thing here is atomic, so atomic size. And with all of these things, it relates if we can see them with the human eye or if we need a light microscope or an electron microscope to view them. Now in terms of cells, the largest cell is the frog egg. And for this, we can just barely see it with the human eye. The next largest cell is the human egg, and the human egg, not quite can we see it with the human eye, but we can see it well with a light microscope, as well as the other types of cells we can see with the light microscope. So cells are very small, and if you have living organisms that are larger than a single cell, then they're made up of many cells. These are multicellular organisms. And that led scientists to a question: why are cells so small? So why are they so small? Why don't we have one cell that is the size of a mouse, or a cell that is the size of a human, or even one individual cell that is the size of a blue whale? The answer to the question as to why cells are so small can be answered when you look at a mathematical relationship between surface area and volume. So surface area. So if you have a cell, the surface area is the membrane. So it's the surface of that cell. And with the surface area, this is where you have all the interactions with the environment. So interactions with the environment, that's what relates to the surface area. Then when you compare this to the volume is everything inside the cell. So the volume, this is the biochemistry, the metabolism that's going on inside the cell. So if you think about it in terms of function, the surface area is the interaction with the environment. The volume indicates the amount of metabolism that is going on in the cell. So remember, with metabolism, you have catabolic reactions and anabolic reactions. So that involves macromolecules. And how do those macromolecules get into the cell? That is through the surface area. So the macromolecules have to pass into the cell, then they are used in metabolism, and then waste products come out. So for this, as a cell increases in size, and you can use cubes to demonstrate this because the math is very easy with the cubes. If you have a cell that is a 4 centimeter cube, so this is the entire cell, then the total surface area is 96 centimeters squared, the total volume is 64 centimeters cubed, and then the surface area is 1.5 to 1. So that's how much surface area you have compared to the volume. Now, if you take that cube and divide it into 8 smaller cubes and calculate the surface area and volume, what you see is that the surface area is 192 centimeters squared, the volume is 64 centimeters cubed, and now the surface area is 3 to 1. So now you have twice as much surface area compared to the volume. And if you make those cells, those cubes, even smaller, you divide it into 64 1 centimeter cubes, then the total surface area is 384 centimeters squared. Volume is 64 centimeters cubed, and now it's 6 to 1. So as the cells get smaller, that increases the amount of surface area compared to the volume. So what that does is as the cells get smaller, that increases the amount of interactions with the environment, the ability of the cell to get nutrients and to release waste products compared to the metabolism. So small cells are able to support their metabolic processes better than larger cells. If you had a very large cell, then the cell would not be able to take in enough nutrients or to get rid of the waste compared to the metabolism that's going on. So that is why cells need to be very small so that their interactions with the environment can support the metabolism that is going on inside the cell. Because of the importance of the surface area to volume ratio for cell functioning, most cells are too small to be seen with the naked eye. So in order to see them, we need to use microscopes. This is an example of a light microscope. Light microscope has a light source which passes light through the sample and then uses lenses, objective lenses and ocular lenses to magnify that sample. This is an example of what you could see for some cells using a light microscope, and these are individual cells. Also, there are electron microscopes which are used to view even smaller structures. This is an example of a scanning electron microscope used to view particles of pollen. And this is an example of a transmission electron microscope which can view the inner structures of one particular cell. In class, we will be using light microscopes to view cells. As the cell theory states, all organisms are composed of cells. And there are two basic types of cells, prokaryotic cells and eukaryotic cells. And in general, prokaryotic cells are very small cells, they're simple in structure, and they do not have any organelles. And organelles are these membrane-bound areas inside the cell. Eukaryotic cells, on the other hand, are much larger, they're more complex, and they do have organelles. They do have these membrane-bound areas inside. Now, back when they first developed microscopes and started to look at cells, which remember most cells cannot be seen with the naked eye, what they noticed with these two cells is that you had very small cells with no membrane-bound organelles, and then you had these large cells that had these organelles or these membrane-bound areas. And they developed these names during this time, and eukaryote, eu means true, karyo means kernel. So they call these cells the true kernel cells. And then the smaller cells they called prokaryotic cells, pro meaning before, karyo meaning kernel. Because what they thought at the time was that these small cells were going to develop into these larger cells and develop this kernel, which we know now is the nucleus. So that's what they thought at the time, and that's where these names come from. The prokaryotic, pre-kernel cell would then mature into the true kernel cell, the eukaryotic cell. Now we know this is not true. These are two separate types of cells. Prokaryotes do not become eukaryotes, but that's where the names came from. Now going through the different domains of living things, these two types of cells relate to the domains. So prokaryotic cells make up the group bacteria and the group archaea. Eukaryotic cells make up the group eukarya. So the first type of cell I'm going to discuss are the prokaryotes, and the prokaryotes make up the domain archaea and bacteria. So the first one I'll go through are the archaea. Archaea are unicellular organisms, so that means that one individual cell is an individual organism. And most scientists believe that the first cells on Earth were probably archaea. An interesting thing about the archaea is that they tend to live in extreme environments. So a lot of archaea are found in hot springs. They are able to survive in boiling temperatures. They can also be found in high salt environments. They can be found in very acidic environments or very basic environments. So to the point where most archaea are considered extremophiles. They live in extreme environments. Now that means that we, as humans, don't encounter them very often, but the archaea are very interesting to study, and a lot of exobiologists believe that if we find life on other planets or out in outer space, they'll probably be similar to the archaea because they will be adapted to these extreme environments. The other type of prokaryotic cell are the bacteria. And bacteria are found everywhere. They can be found in the air, in the water, in the soil. They're on the surface. They've colonized the surface of all living things. So you have bacteria covering the whole surface of your skin. And in general, bacteria come in three different shapes. They can be in a rounded shape, like a spherical shape, and these are called cocci. Or they can be in rod-shaped, and this is called bacilli. Or they can be in spiral shape, and that is just called spiral. So there are hundreds of thousands, maybe millions of different species of bacteria, and of all those bacteria, or all those different species of bacteria, they'll come in one of these three shapes: cocci, bacilli, or spiral. Now I want to go through the basic structure of a bacterial cell. So again, bacteria are unicellular organisms, so one individual is composed of one cell. This example shows a bacillus bacterial cell, so remember bacillus is rod-shaped. Bacterial cells are very simple in structure, but I'm going to go through it systematically. I'm going to start with the appendages on the outside of the cell. Then I'm going to talk about the layers of the cell, and at the end I'm going to talk about what you find inside a bacterial cell. The first appendage that I want to talk about are the flagella, the bacterial flagella. And flagella means whip, and these are long appendages, usually very long compared to the size of the cell, and very thin. And flagella are used for locomotion. So when bacteria locomote, they run in a straight direction, and then they sort of tumble around, turn around, and then they run in a straight direction, and then tumble around, and then run. So that's how bacteria locomote. And only bacilli and spiral bacteria can have flagella. Flagella can also be used for identification because there are different arrangements of flagella on bacteria. Peritrichous bacteria can have flagella all over the surface, evenly distributed over the surface. Monotrichous, mono means one, it has one polar flagella. Lophotrichous, lopho means a tuft, where it has several polar flagella. And amphitrichous, amphi means both, it has flagella at each pole. So again, flagella are used for locomotion, and only bacilli and spiral bacteria may have flagella. The second type of appendage that you may find on bacteria are fimbriae, and fimbriae are these numerous extensions that come out of the surface of the bacteria, and they are used for adherence. So they help the bacterial cell adhere to surfaces. The third type of appendage that bacterial cells may have are called pili, pilus for singular. And a pilus is an appendage that connects two bacteria to each other. And the purpose of connecting the two bacteria to each other is to transfer DNA from one bacterium to another. This process of DNA transfer is called conjugation. So this is the process where a donor bacterium can form a pilus connecting it to a recipient bacterium, and then across that pilus a copy of DNA will be transferred, and that DNA molecule is called a plasmid. So during conjugation, a pilus is formed connecting two bacteria, and a copy of the plasmid is transferred to the recipient bacteria. So at the end of the process, both bacteria have a copy of that extra plasmid. So I went through the three types of appendages that bacterial cells may have: flagella, fimbriae, and pili. Next, I want to start going through the different layers that bacterial cells have. The first layer you may find on a bacterial cell is called a glycocalyx. Glyco means sugar, calyx means coat. So this is an outer layer of carbohydrates. And remember, carbohydrates are made of simple sugars. Those are the monomers that make up the polymer of carbohydrates. So simple sugars, glyco means sugar. One type of glycocalyx is called a slime layer. Slime layers are very loosely held around the cell, and they are disorganized, not very organized. But the purpose of the slime layer is attachment. So think about sugar. When sugar gets wet, it gets very sticky. So that's the purpose of the slime layer. And this is found mainly on non-pathogenic bacteria, bacteria you find in the environment. Second type of glycocalyx that bacterial cells may have is called a capsule. And capsules are held very tightly around the bacterial cell, and they're very organized. So if you look at their molecules, the carbohydrates, they're very, very organized. And the purpose of a capsule is also attachment, but capsules are found mainly on pathogenic bacteria, bacteria that can infect and cause diseases in humans. And the capsule can also protect the bacteria from your white blood cells. So it actually makes the bacteria more pathogenic because it's harder for your body to fight off an infection of bacteria if they have a capsule. The outermost layer of a bacterial cell is the glycocalyx. As you move into the cell, then we would come in contact with the cell wall. The purpose of the cell wall is to hold the shape of the bacterial cell, so to hold its shape, whether it's a coccus, bacillus, or spiral. Second purpose of the cell wall is to protect from osmotic lysis, to prevent the cell from rupturing because of the movement of too much water into the cell. Now, bacterial cell walls have two major types: gram-positive cell walls and gram-negative cell walls. And gram just refers to the scientist who identified the staining procedure, which allows us to distinguish between the two types. Now, the important thing about the bacterial cell wall is that bacterial cell walls contain peptidoglycan. So peptidoglycan is there and peptidoglycan is there. So whether it's a gram-positive cell wall or a gram-negative cell wall, both of them contain peptidoglycan, which is not found in eukaryotic cell walls. The last layer that you find in a prokaryotic cell is the cell membrane. So this is the most interior layer. So again, outermost layer is the glycocalyx, then the cell wall, then the cell membrane. And the purpose of the cell membrane is to separate the interior of the cell from the environment. Also, another purpose of the cell membrane is transport. So it should allow nutrients to go into the cell and waste products to go out. So cell membranes are semi-permeable. They will allow certain molecules to move into the cell and other molecules to move out of the cell. Basic structure of the cell membrane is the phospholipid bilayer. So this is the phospholipid bilayer with those phospholipids in two layers. Each phospholipid has a polar head that interacts with the water outside of the cell and inside of the cell. And then the non-polar tails or hydrophobic tails are forced together in the interior of the membrane. So the phospholipids make up the main matrix of the membrane, but also embedded in the membrane you have all sorts of different proteins. So different types of proteins. Some of these proteins are channels which allow molecules to move into or out of the cell. And in a prokaryotic cell, a lot of these proteins embedded in the membrane act as enzymes and catalyze reactions for the cell. So far, for the structure of the prokaryotic cell, I've gone through the 3 different types of appendages, the 3 different layers of the prokaryotic cell, and now what I want to do is go through what you find inside the cell. The first thing that you would find is a chromosome. And a chromosome in a prokaryotic cell is located in an area called the nucleoid. So this is the area of the cell where you find the nucleoid. Now the nucleoid is not an organelle because prokaryotic cells do not have organelles. Now the chromosome of a prokaryotic cell is in a circular shape. So it is a circular piece of DNA that is composed of DNA only. And every prokaryotic cell has only 1 chromosome. Ribosomes are also found inside prokaryotic cells, and a ribosome is a complex of RNA and protein. So that is what a ribosome is composed of, RNA and protein, and there are 2 subunits, a small subunit and a large subunit. And ribosomes are responsible for producing proteins, performing translation. And prokaryotic ribosomes have a particular measure. They are measured as 70S, which means they are smaller and less dense than eukaryotic ribosomes. Eukaryotic ribosomes have a measure of 80S. So the point of this is you can identify prokaryotic ribosomes from eukaryotic ribosomes. The last thing that you may find inside a bacterial cell are plasmids, and plasmids are small circular pieces of DNA. And these plasmids are transmitted across a pilus during conjugation. And depending on how often a bacterial cell has conjugated, one particular bacterial cell may have 0 plasmids, or it could have hundreds of plasmids, depending on, again, how often it has participated in conjugation. And plasmids can be important because they're often used in biotechnology, and they often have antibiotic resistance genes on them. As a summary for the basic structure of a prokaryotic cell, starting with the appendages, some bacterial cells may have a flagella for locomotion. They may have fimbriae, which will improve attachment, and they also may have a pilus, which will facilitate conjugation. As for the layers of the bacterial cell may have a glycocalyx. In this case, it's a capsule, which will help with adherence to surfaces and also protect the bacterial cell from your white blood cells. All bacterial cells will have a cell wall. The cell wall holds the shape of the cell. In this case, this is a bacillus, a rod-shaped bacteria, and it also prevents osmotic lysis. The last layer that you will find on all the cells is a plasma membrane, and the plasma membrane separates the inside of the cell from the environment, and it also facilitates transport of nutrients into the cell and waste products out of the cell. Now, inside the cell, you're going to find the nucleoid. The nucleoid is the area where you find the chromosome. And every bacterial cell has only 1 chromosome. It is circular in shape and composed of DNA only. Also, inside the bacterial cell, you're going to find ribosomes. These ribosomes are very important because they are going to produce the proteins for the bacterial cell. And prokaryotic ribosomes measure 70S. The last thing you may find in a bacterial cell is a plasmid. A plasmid is a relatively small circular piece of DNA, much smaller compared to the chromosome, and this is a result of conjugation where the plasmid was transferred across a pilus into a recipient cell. And plasmids, again, may or may not be found in a bacterial cell depending on how many times it has conjugated. Second type of cell I want to discuss are the eukaryotic cells. And remember, U is true, karyo means kernel. So these are the very large complex cells that have that kernel inside that is visible with a light microscope. And now, of course, we know the kernel is the nucleus. And eukaryotic cells make up the domain of all the organisms in eukarya. So that's pretty much every organism besides the archaea and the bacteria. So that makes up all of the animals and also the plants. Eukaryotic organisms can be unicellular, like the protists, amoebas. An individual cell makes up an individual. Or they can be multicellular organisms, like humans or any animal and plants. And those are multicellular. They're made of thousands, trillions of cells. Like the prokaryotic cell, I'm going to start from the outside of the cell, talking about the appendages, then the layers of the cell, and then moving on to the interior of the cell. There are two types of appendages that eukaryotic cells may have. First are flagella, flagellum for singular, and flagella, like in prokaryotic cells, it's used for locomotion, getting the cell from one place to another. And while it serves the same purpose, they do have slightly different structures, prokaryotic flagella and eukaryotic flagella. The second type of appendage are cilia. And cilia are only found in eukaryotic cells. Prokaryotic cells do not have cilia. And the cilia can also be used for locomotion in unicellular organisms, or it can be used to move fluids in multicellular organisms. Cilia can be found on unicellular organisms, and when it is found on unicellular organisms, it is used for locomotion, but it can also be part of multicellular organisms. In humans, cilia can be found in the trachea, on the ciliated epithelial tissue, and this is the tissue. Tissue is made up of many cells. Each darker spot is a nucleus. And here is the cilia, sort of looks like shag carpeting on top of those cells. And the purpose of the cilia in a multicellular organism is to move a fluid. And in this case, it's moving mucus. So this ciliated epithelial tissue is found in the trachea. When a person inhales air through the mouth or the nose, that air contains particles that will be trapped in the mucus. So trapped in the mucus, and then the cilia, so these are the cilia extensions, they're going to move, and they're going to move the mucus away from the lungs. So that ciliated epithelial tissue is going to move the mucus away from the lungs to prevent those particulates from infecting the lungs. Now I want to go over the different layers of a eukaryotic cell. The outermost layer that you find on most eukaryotic cells are the cell walls. And these are found in all eukaryotic organisms except for animal cells. So that is the exception. And the purpose of the cell wall is to hold the structure of the cell. And most of the cell walls that you find, especially in plants, the major component is cellulose. So cellulose, which I discussed in the macromolecule lecture, that is used for the structure of the plants and the structure of the cell wall. As you move internally to a eukaryotic cell, from the cell wall, you will encounter the plasma membrane. All living cells, prokaryote and eukaryote, must have a plasma membrane. And it serves the same function in the eukaryotic cell as it does the prokaryotic cell, and that is a semi-permeable barrier. So it is a barrier which separates the inside of the cell from the environment, but it's semi-permeable, so it should allow transport of molecules into the cell, such as nutrients, and transport of some molecules out of the cell, such as waste products. And like the prokaryotic cell, the main matrix is the phospholipid bilayer. So again, phospholipids with the polar heads interacting with the water inside the cell and outside the cell, and then the nonpolar tails. There are two differences between a eukaryotic cell membrane and a prokaryotic cell membrane. First is that eukaryotic cell membranes contain cholesterol. So inside that phospholipid bilayer, in eukaryotic cell membranes, you will have cholesterol. The other difference is that in eukaryotic cell membranes, these proteins that are embedded into the cell membrane are usually not enzymes. So the enzymes are found mainly in the organelles in a eukaryotic cell. In prokaryotic cells, because prokaryotes do not have organelles, they have a lot of enzymes embedded in the membrane. I've gone through the appendages of a eukaryotic cell. They can have flagella or cilia. I've also gone through the two layers of a eukaryotic cell. They may have a cell wall, and then they all must have a plasma membrane. So now I want to discuss what you find inside a eukaryotic cell. And before I go through that, I just want to distinguish between two terms, cytoplasm and cytosol. And sometimes these are used interchangeably, but they're not interchangeable. So cytoplasm refers to everything you find inside the cell. So that would be everything that would be the liquid solution you find inside the cell and the different organelles you find inside the cell. Cytosol only refers to the liquid that you find inside the cell. So when you're referring to that liquid solution, technically you should refer to it as cytosol. One thing you will find inside a eukaryotic cell is a cytoskeleton. And the cytoskeleton is a network of protein fibers, 3 different types of protein fibers. You have intermediate filaments, microtubules made of tubulin, which are very large, and then the microfilaments made of actin, which are very small. So this network of protein fibers has a couple different purposes. One is to hold the shape of the cell, especially in animal cells that do not have a cell wall. The other thing that the cytoskeleton does is it holds the organelles in place. So most of these organelles are not just free-floating inside a eukaryotic cell. And the third thing the cytoskeleton does is it facilitates movement inside the eukaryotic cell. So eukaryotic cells may have a cytoskeleton, prokaryotic cells do not. I mentioned that the cytoskeleton is involved in movement, and there are 2 types of movement that it can be involved in. First of all, the cytoskeleton is involved in the structure of flagella and cilia, so it facilitates locomotion or movement of liquids around the cell. But the cytoskeleton is also involved in cytoplasmic streaming. So cytoplasmic streaming, this is the movement of the cytosol actually. The cytosol circulating through a eukaryotic cell. Now eukaryotic cells are very, very large. In the cellular world, they're large cells. And they cannot wait for just simple diffusion to move a nutrient from one side to the other. So there's a lot of metabolism going on in a eukaryotic cell, and they need to be able to move the nutrients around very quickly. So by contracting the different fibers in the cytoskeleton, that facilitates movement or circulation of the cytosol. Also, cytoplasmic streaming can be responsible for amoeboid movement. Amoeboid movement, that's using pseudopods. So pseudopods where the cell membrane extends out and the cell crawls around. That's how amoebas move. And this is also caused by cytoplasmic streaming, where the cytosol sort of fills up an area of the cytoplasm and it bulges out, and that allows the cell to crawl around. So the cytoskeleton is involved in movement with the flagella and the cilia, and also cytoplasmic streaming, which facilitates movement of the cytoplasm around the interior of the cell to distribute nutrients, and also amoeboid movement. Another structure related to the cytoskeleton is the centrosome. And the centrosome is composed of 2 centrioles and this is an actual electron micrograph of a centrosome or two centrioles. And these two centrioles, or centrosome, is referred to as the MTOC, microtubule organizing center. So this is a structure that organizes microtubules. And this is very important during mitosis. So mitosis is the process of cell division, and during this process, all the chromosomes are copied in the eukaryotic cell, and then those two sets of chromosomes need to be separated from each other. So these two sets of chromosomes are the purple structures here, the dark purple, dark blue, and the microtubules are all these pink fibers. And the pink fibers, the microtubules, have attached to the chromosomes, and then they pull the two sets of chromosomes apart, so then the cell can divide, and then each daughter cell will have its own set of chromosomes. In addition to the cytoskeleton, eukaryotic cells will have organelles, and that's a defining characteristic of the eukaryotic cells compared to the prokaryotic cells. And a basic definition of an organelle is a membrane-bound area of cytoplasm that has a specific shape and function. So three key components. It's membrane-bound, it has a specific shape, and a specific function. And the reason it has a specific function is because certain organelles contain certain sets of enzymes. Now I'm going to go through the typical organelles that are found in eukaryotic cells. And always we seem to begin with the nucleus, probably because it is the largest organelle and the easiest one to see. So this is the nucleus. The purpose of the nucleus is to protect the DNA. So inside the nucleus you will find the chromatin, which is the DNA. Often you will also see a nucleolus. The nucleolus is not an organelle. It is a darker staining area in the nucleus. And the nucleolus is where the rRNA is made. The rRNA, this is the RNA that makes up part of the structure of the ribosome. So that's the nucleus. Now the nucleus also has pores. It's a double membrane and it has pores opening in it. And that will allow some molecules to come out, like messenger RNA. So messenger RNA can come out, and it can also allow proteins to go in. So proteins can go in. But DNA never leaves the nucleus. The whole purpose of the nucleus is to protect the DNA. When comparing the chromosomes of prokaryotic and eukaryotic cells, there are several important differences. The first difference is that in eukaryotes, there are multiple chromosomes. Multiple. So for instance, humans have 46 chromosomes, whereas prokaryotes only have 1. Another difference between eukaryotic and prokaryotic chromosomes is their shape. Eukaryotic chromosomes are linear, whereas prokaryotic chromosomes are circular. And the last difference is that eukaryotic chromosomes contain proteins called histones. So actually a eukaryotic chromosome is a complex of DNA and protein, whereas the prokaryotic chromosomes were composed of DNA only. The next organelle that is usually discussed is the endoplasmic reticulum, or the ER. And the ER, the endoplasmic reticulum, is actually an extension of the nuclear envelope. So if you look at this diagram, the nuclear envelope is in gray, and as it extends out, it forms the green ER. So the ER, endoplasmic reticulum, is very closely associated with the nucleus because it extends out from the nuclear membrane. There are two types of ER. There is the rough ER, which is right next to the nuclear envelope. And this is called the rough ER because it contains ribosomes. So those ribosomes have the same purpose as prokaryotic ribosomes. They are used to make protein. And ribosomes, eukaryotic ribosomes, are also a complex of RNA and protein. And they also have two subunits, a large subunit and a smaller subunit, but overall the ribosomes are larger, so they measure 80S. So again, you can identify the differences between a prokaryotic ribosome and a eukaryotic ribosome. So because ribosomes are found on the rough ER, the purpose of the rough ER is to produce proteins. The other type of ER is called the smooth ER, and the smooth ER is called smooth ER because it does not have ribosomes embedded in the membrane. And the purpose of the smooth ER can be to produce lipids and some carbohydrates, so it has enzymes that can do that. And also the smooth ER can detoxify the cell. So any sort of toxic substance that enters the cell can be inactivated by enzymes found on the smooth ER. Going over ribosomes again, ribosomes are essential for living cells because they produce proteins, and all living cells need to produce proteins, especially those that act as enzymes, which will facilitate metabolism. And prokaryotes and eukaryotes both have ribosomes. Those ribosomes are composed of two subunits, a large subunit and a small subunit, and both subunits are composed of RNA and protein. And in eukaryotes, that ribosome measures 80S, and they can be found bound to the rough ER or free-floating in the cytoplasm. The next organelle is the Golgi, sometimes referred to as the Golgi apparatus or the Golgi complex. And the Golgi is a series of flattened sacs that is found closely associated with the ER. And the purpose of the Golgi is to take the products of the ER, to modify them, and to package them and deliver them to other parts of the cell or outside of the cell. So the products of the ER will be transported to the Golgi. They'll go through a process in the Golgi where they are modified and then packaged into transfer vesicles or secretory vesicles, and then they are transported to other parts of the cell, or those products of the ER are released into the environment. Lysosomes are organelles that contain digestive enzymes. So these are organelles that are composed of a vesicle, membrane-bound area that contain the digestive enzymes. And there are a couple different purposes for the lysosome. One of the major purposes is in phagocytosis. When the cell takes in a food particle, nutrients, it will form a vesicle around that nutrient called a food vacuole, and then that food vacuole will fuse to a lysosome so that those digestive enzymes in the lysosome can then interact with the food particle and digest it into smaller subunits that the cell can then use. Also, lysosomes are involved in autophagy, auto-self-phagy to eat, and the lysosome can be used to digest and break down old organelles that are not functional anymore. And lysosomes can also be involved in apoptosis, which is a type of cell death, because if the lysosome is ruptured, so if for some reason this membrane is ruptured, then these digestive enzymes can go into the rest of the cell and digest the nucleus, the ER, the Golgi apparatus, and everything, resulting in the death of the cell. So sometimes apoptosis is known as cell suicide. The organelles I have discussed so far are part of something we call the endomembrane system. And it's called the endomembrane system because they share a membrane that can fuse together or bud apart, and also they are part of a system. They all work together. So as an example of this endomembrane system, you would start with the nucleus. The nucleus contains the DNA that encodes the information to make types of proteins. So for instance, there is a gene in the nucleus that encodes the information to make a digestive enzyme, which is a protein. That information will leave the nucleus. It will go to the rough ER, where it will interact with ribosomes on the rough ER. The rough ER, or those ribosomes, will produce the protein. Protein will then be transported to the Golgi apparatus. So it's transported to the Golgi apparatus where that protein, that enzyme, will be modified and packaged into a vesicle which will form the lysosome. So the lysosome will then serve to digest different food particles that have entered the cell. So that's one example of the endomembrane system where those organelles are working together. There are a few more organelles that I wish to discuss, and the first one are the peroxisomes. And the peroxisomes are very similar to lysosomes. They are membrane-bound vesicles that contain enzymes, and these enzymes are associated with breaking down fatty acids specifically. Vacuoles are another type of organelle that you can find in eukaryotic cells, and the most common types of vacuoles are found in plant cells, and they are used for storage. Often when you look at a plant cell, you'll see a very large vacuole called the central vacuole, which is storing water for the plant. Also, some protozoa have vacuoles, contractile vacuoles that store water, and then when the protist contracts it and eliminates the water, it helps with motion for that protist. Another type of organelle that are found in plant cells are the plastids. There are three basic types of plastids. Leucoplastids are used for storage, carbohydrate storage and/or oil storage. There are chloroplasts, which are used for photosynthesis, and then chromoplasts, which contain pigments. The last two organelles I want to discuss are energy-related structures. The first one are the chloroplasts. Chloroplasts take light energy and convert it into glucose. And the second type of organelle are the mitochondria, which take the glucose and convert it into ATP. So these are the energy-related structures. Converting light into glucose is the process called photosynthesis, and taking glucose and converting it into ATP is called cellular respiration. Now, there are a couple of interesting things that these two organelles share. Both of them have two membranes. They have an outer membrane and an inner membrane. They both have their own ribosomes, and the ribosomes measure 70S, and they both contain a circular molecule of DNA. I mentioned at the beginning of the lecture that scientists believe that the very first cell that arose on Earth was probably an archaea, a type of prokaryotic cell. So this was a very small cell with a cell wall and no organelles. And they believe that over time, a couple million years, that cell evolved into a eukaryotic cell, which is a much more complex cell with lots of organelles. Now, the idea of how a prokaryotic cell, or how prokaryotic cells evolved into eukaryotic cells, is a process called endosymbiosis. Endo meaning inside, symbiosis living together. This is a diagram of how scientists believe endosymbiosis occurred, or how eukaryotic cells evolved. So first, what they believe happened is that you start with a cell very similar to an archaea, and the cell wall was lost. And this is important because you have to have movement of the cell membrane. So the cell wall was lost, and then the membrane started to fold infolding, and it started to surround the DNA. So it started to surround the DNA, and that is the beginning of the formation of the nuclear envelope. So the nuclear envelope, and at the same time you had cytoskeletal fibers forming. So as this continued on, the DNA is surrounded by a membrane forming the nuclear envelope, and then the extension of that membrane forms into the ER, the rough ER and the smooth ER. Then over time, that could form into the Golgi apparatus. So parts of that membrane could separate off, forming the Golgi apparatus, that series of membranous sacs. Then last part that would have happened is that at some point the cell would have engulfed a smaller cell, and normally the purpose of that would be to digest it, to eat it and use it as nutrients, but at some point it evolved a symbiotic relationship. That's where symbiosis comes in, And now that little bacterial cell evolved into the mitochondria, producing ATP for the larger cell. And then later over time, the cell may have engulfed a photosynthetic bacteria, and now that performs photosynthesis for the larger cell. A little bit more detail on the endosymbiosis part of this eukaryotic cell evolution. Endo inside, symbiosis living together. So this really refers to the development of the mitochondria and the chloroplast. So the idea again is that at some point in the development of the eukaryotic cell, the cell would have phagocytosed a bacterium that can perform cellular respiration, and eventually, instead of digesting that bacterium, they worked out a symbiotic relationship where that bacterium would then produce ATP for the cell. In terms of the chloroplast, that cell at some point would have phagocytosed a photosynthetic bacteria or a prokaryote, and then again, instead of digesting it, they would have worked out a symbiotic relationship where that prokaryote would have produced glucose from photosynthesis for the cell. Now the evidence for this. Where is the evidence for this idea? The first piece of evidence is the fact that it has two membranes. The mitochondria and the chloroplast have two membranes. And this would come from that process of phagocytosis. So if you have a larger cell that's going to phagocytose a smaller cell, what will happen is the larger cell has to move the membrane to form a vesicle around the smaller cell. So what you will end up with is once that smaller cell is engulfed and brought into the larger cell, you're going to have a structure that has two membranes. That vesicle membrane that was formed during phagocytosis, and then the original prokaryotic cell. So that would be evidence of two membranes. And in fact, in the mitochondria and the chloroplast, the outer membrane for those structures is very similar to a eukaryotic membrane, and the inner membrane is very similar to a prokaryotic membrane, so they can tell the difference. The other evidence for this process is the fact that mitochondria and chloroplasts have their own ribosomes, and those ribosomes measure 70S. So I pointed out a couple of times that you can tell the difference between a prokaryotic ribosome and a eukaryotic ribosome by this measurement of 70S. So these two organelles contain prokaryotic ribosomes. And the last piece of evidence is that both of these organelles have a small circular piece of DNA. So they each contain a small circular piece of DNA, pure DNA, which looks just like a prokaryotic chromosome. So these are physical signs or evidence that at some point in evolutionary history that the mitochondria and the chloroplasts were once free-living prokaryotic cells that were engulfed by a larger cell, and they worked out a symbiotic relationship. As a review for the structure of eukaryotic cells, I'm going to use this image, which is sort of a fusion of a plant cell and an animal cell. So I started with the different types of appendages. Eukaryotic cells may have a flagella or cilia. Then the different layers of the cell, they may have a cell wall like plant cells, but they all must have a plasma membrane. So those are the layers of the cell. Then, starting with the organelles of the endomembrane system, start with usually the largest, most visible organelle, which is the nucleus. Nucleus contains the DNA, which contains the information to make different proteins. Extending out from the nuclear membrane is the ER. The rough ER has ribosomes, which will serve to translate that information into the proteins. Then you also have the smooth ER, which does not have ribosomes, but can synthesize lipids and carbohydrates and detoxify the cells. Closely associated with the ER is the Golgi complex or the Golgi apparatus, series of sacs, which will take the products of the ER, modify them, package them into vesicles, and then deliver them to different parts of the cell. So delivering them to different parts of the cell, that can also create the lysosome. Lysosome is a vesicle that contains digestive enzymes that can digest food particles or old organelles, or if they rupture, they can cause the death of the cell. Also, some other organelles, peroxisomes, you have peroxisomes, vacuoles, and other different types of plastids. Organelles associated with energy would be the chloroplasts, which perform photosynthesis, and then the mitochondria, which perform cellular respiration. In addition to the organelles, you also have the microfilaments, microtubules, and intermediate fibers, which form the cytoskeleton. And then you have the centrosome, which organizes the microtubules. So those are the basic structures of a eukaryotic cell.